Begin your experiment on the Rosalind homepage. Here you can navigate through your created projects, spaces, and public showcases. Projects are personal places for you to store and organize experiments and meta-analysis. Select Add New Project to create a new green project folder. Give your new project a title and optional description. Both the title and description can be edited at a later time from the homepage. Once you have completed naming your project, hit Next. Select Design a new experiment to access RNA sequencing assays and other sequencing assays. Now give your new experiment a title and optional description. Similar to projects, both the title and description can be edited at a later time. Once you have completed naming your experiment, hit Next. Tell Rosalind your technology used. For RNA sequencing, we will select Sequence. Then select your experiment type from the multiple processing options available within Rosalind. For RNA sequencing, Rosalind can process files from both FASTQ and processed counts. If creating an RNA sequencing experiment from processed counts, select Processed Counts. This will take you to a page for counts file upload in CSV or text format. Use the toggle to tell Rosalind if your processed counts are raw or normalized. Then select the blue File Upload box to navigate to your counts file or drag and drop. Optionally, you can upload an attributes file at this stage, but we will demonstrate this at a later point in experimental setup. Once your file is uploaded, hit Next. Now we will show you how to upload from FASTQ files. From the previous experiment type upload screen, select RNA sequencing FASTQ. Experimental setup will now be nearly identical between both file types. Next, define your species from the many genomes available for analysis. Then select your genome build from NCBI or Ensembl. We recommend to select the most recent genome for your species. Record your sample and library kit used during your experiment by selecting the kit vendor and model. If you do not see your kit in our selection, you can reach out to our support team for further guidance, or select Other to run a generic unstranded analysis. Once you have made your selection, click to Add Kit and ensure you have both a sample and library kit added to continue on to the next step. Note if your experiment has biological replicates. This setting can be useful for tracking sample replicate information, which will be shown later. Then input your total number of samples. Enter your study attributes by manually typing in the name of your attribute. Rosalind Intelligence will bring up closely matching attributes from the Rosalind and NCBI databases. You can select a matching attribute from the drop-down menu or hit Enter to create your own attribute. Once you have added all of your study attributes, select Generate Attribute File to download a CSV file of your created attributes. You can enter your sample attribute information in the CSV or sample by sample later on within the Rosalind platform. Within the CSV file, enter a unique sample name for each sample. Then fill in the corresponding information related to each sample's attribute. Once all the attribute information is completed, save the file as a CSV for re-upload into Rosalind. The attribute file can be re-uploaded by selecting Upload Attribute File. You can select Browse or Drop File to navigate into your files for selection, or drop your file into this box from a separate window. Once the file is uploaded, you will see the total number of attributes from the file on the right-hand side. If this looks correct, select Next to confirm which attributes were detected. Review your final sample information. Optionally, you can input laboratory identifier and tube plate barcode numbers for internal tracking within experiments. You can remove samples from your final list by selecting the red minus on the right-hand side. You can add additional samples from the bottom Add Samples button. If your sample information is approved for analysis, select Next. Assign which samples are biological replicates. Select the drop-down menu for each sample and select which sample is a replicate of that sample. Once you select a replicate, you will see a corresponding color change. All replicate groups will have a unique color for easy visual tracking. If a sample is placed into the wrong replicate group by mistake, the mismatched colors should alert you to make a correction. Assigned replicate groups can assist with final sample review. If any changes need to be made to your attributes at final review, changes will be applied to all samples within their replicate group automatically. For example, here we are changing the time course from 48 hours to 12 hours. This change in one sample now is automatically applied to all 3 samples within this replicate group, reducing time required for editing as well as risk of technical errors. Final sample review can be performed sample by selecting Next, or skipped by selecting the last sample red dot on the right. You can view your final sample information as a standard table, or by replicate group colors by selecting the top drop-down Colored by menu and selecting Replicates. After all samples and information is confirmed, select Yes, I have reviewed my sample data. Comparisons can be created during experimental setup or later on after the experiment has been launched. To create a new comparison during experimental setup, select Create New Comparison. You will see gray selection boxes depicting your study attributes and individual samples. Comparisons can be set up from the attribute or sample level. To create your comparison, select your attribute of interest and drag and drop that attribute into the corresponding box for control or condition. You will notice that your default comparison name will intelligently change as you drag attributes into boxes. Rosalind is also able to detect if your experiment has appropriate attributes for a covariate correction. You can optionally select to perform covariate correction within your comparison to correct for confounding variables. When you are satisfied with your comparison, select Save. You are able to set up multiple comparisons for differential analysis. To consider multiple factors, such as a treatment at a single time point, simply drag and drop multiple attributes into their corresponding boxes. Notice that the default name of the comparison changed to reflect this as well. The last step in experimental setup is to upload your FASTQ files. Rosalind can process FASTQ, GEZ, or GZIP formatted files. Use the toggle to select your sequencing file type, whether single-end or paired-end files. Notice that the blue sample file upload boxes change for each option, as paired end require a left and right read per sample. For this example, we will use single end. To upload your FASTQ files, either select the blue sample upload box to navigate to your files or drag and drop your files from a separate window. Notice that the file will immediately begin uploading. Once all sample files have been uploaded, your experiment will automatically launch. Do not navigate away from the upload page until all files have been uploaded and your experiment is launched. Thank you for watching.